Let's discuss about the types of necrosis. There are several types of necrosis as it can affect many areas of the body, including bone, skin, organs, and other tissues. Each type has distinct features. The type of necrosis that develops depends on which tissues are necrotic and what events are triggered it. The types include caseous necrosis, fat necrosis, fibrinoid necrosis, coagulative necrosis, liquefactive necrosis, and gangrenous necrosis. Now, the number one is coagulative necrosis. It is a form of tissue necrosis in which the component cells are dead, but the basic tissue architecture is preserved for at least several days. The affected tissue takes on a firm texture. Pre-assembly, the injury denatures not only structural proteins but also enzymes and so blocks the proteolysis of the dead cell. As a result, eosinophilic anucleate cells may persist for days or weeks. Most common type of necrosis is coagulative necrosis and mostly from sudden cessation of ischemia, less often from bacterial and chemical agents. It's a characteristic of infarct, which is called areas of ischemic necrosis in all solid organs except the brain. The organs commonly affected are the heart, kidneys, and spleen. If we talk about its gross appearance, it has a pale segment may be seen in contrast to surrounding healthy tissue. The segment may be hard to touch and the microscopic appearance is in H&E staining tissue. Eosinophilia like cell will be noticeable. Anucleated cells should be observable with preserved cell outlines, and the affected area on right side picture shows cells with the intensely eosinophilic cytoplasm of tubular cells, but the outlines of tubules are still maintained. The nuclei shows granular debris, and the interference between viable and non-viable area shows non-specific chronic inflammation and proliferating vessels. Second type is liquefactive necrosis. Due to ischemic injury and bacteria or fungal infection, degradation of tissue by the action of powerful hydrolytic enzyme. The common examples are infarct, brain, and abscess cavity. Whatever the pathogenesis, liquefaction completely digests the dead cells, resulting in transformation of the tissue into a liquid viscous mass. If the process was initiated by acute inflammation, the material is frequently creamy yellow and is called pus. The gross appearance is the affected area is soft and liquefied center containing necrotic debris. Later, a cyst wall is formed. The microscopically, the cyst space contains necrotic cell debris and macrophages filled with phagocytosed material, and the cyst wall is formed by proliferating capillaries, inflammatory cells, and gliosis in the case of brain, proliferating fibroblasts in the case of abscess cavity. Liquefactive necrosis brain, the necrosed area on right side of the picture shows a cyst space containing cell debris, while the surrounding zone shows granulation tissue and gliosis. Third type is caseous necrosis. It is found in the center of foci of tuberculosis infection. It combines feature of both coagulative and liquefactive necrosis. The term caseous is derived from the friable yellow white appearance of the area of necrosis. Grossly foci of caseous necrosis, as the name implies, resemble dry cheese and are soft, granular, and yellowish. This appearance is partly attributed to the histotoxic effects of lipopolysaccharides present in the capsule of tubercle bacilli, Mycobacterium tuberculosis. Microscopically, the necrosed foci are structureless, eosinophilic, and contain granular debris. The surrounding tissue shows characteristic granulomatous inflammatory reaction. Let's see the picture. This is the caseous necrosis lymph node. There is a eosinophilic amorphous granular material, while the periphery shows granulomatous inflammation. Fat necrosis. Fat necrotic tissue is speckled with small white chalky deposits, and these white spots are formed by calcium and fatty acids that are deposited into the necrotic tissue. Grossly, the fat necrosis appears as yellowish white and firm deposits. Formation of calcium soaps imparts the necrosed fossae firmer and chalky white appearance. Microscopically, the necrosed fat cells have cloudy appearance surrounded by an inflammatory reactions. This is a picture of fat necrosis in acute pancreatitis. The area of white chalky deposits in this picture represents foci of fat necrosis with calcium soap at size line breakdown in the mesentery. Now the fibrinoid necrosis. It is characterized by deposition of fibrin-like material which has the staining properties of fibrin, and it is encountered in various examples of immunologic tissue injury like immune complex vasculitis, autoimmune diseases, Arthur's reactions, arterios in hypertension, peptic ulcers. Gross appearance it is usually not grossly discernible, and microscopic appearance is like an amorphous appearance that is bright pink in an H and E stain. The deposition of fibrinoids are surrounding the blood vessels. Inflammation should be present. Um, and this is the picture of fibrinoid necrosis in an artery in a patient with polyarteritis nodosum. The wall of the artery shows a circumferential bright pink area of necrosis with protein deposition and inflammation. Now the last type is gangrenous necrosis. Gangrenous necrosis does not demonstrate a specific pattern of cell death but is preferably used in clinical practice to describe the condition.